Hello everyone, welcome to QXON. So in this video, you will learn about the molecular basis of skeletal muscle contraction. Under this topic, I will first be discussing two important functional units of the skeletal muscle, namely the sarcomere and the sarcotubular system. And then we will discuss the excitation contraction coupling process in detail. Finally, we will compare smooth muscle contraction with that of skeletal muscle. So here we go. Now let's begin by understanding the organization of skeletal muscle. As you can see in this diagram, the skeletal muscle is made up of many bundles of fibers. Each bundle is called a fasciculus. Now the fasciculus is made up of many muscle fibers or myocytes, and each muscle fiber in turn is made up of many long cylindrical filaments called myofibrils. Now the myofibril is in turn made up of repeating units called sarcomeres. We will look at the structure of the sarcomere in more detail a bit later. So if you examine the skeletal muscle under a microscope, you will see that the individual cells are in the form of long cylinders. So we call them as muscle fibers. Now each muscle fiber is multinucleated. Also, the skeletal muscle does not have the ability to regenerate, which means that if you lose some fibers due to some kind of a disease or muscle damage, then cell division cannot compensate for this loss. But hypertrophy can to some extent, and by hypertrophy we mean that the individual muscle fibers will increase in the size but not in number. Now under the microscope, the skeletal muscle fibers appear striated. This is due to the presence of myofibrils, which in turn are made up of alternate light and dark bands. So under the microscope, they appear like the stripes on a zebra. With that background, let us now focus on the structural components which are important in muscle contraction. Now there are two important functional components in the skeletal muscle fiber which are important for muscle contraction. These are the sarcomere and the sarcotubular system. So let's start by discussing the sarcomere. Now we already know that each myofibril is made up of repeating units which are called sarcomeres. So by definition, the sarcomere is the functional contractile unit of the skeletal muscle which is arranged in repeating units. So it is the smallest level at which the skeletal muscle contracts. Now it is the region between two Z lines. We will shortly look at what these Z lines are. Now the sarcomere is made up of three important types of proteins. The most important being the contractile proteins which actually participate in the contraction, that is actin and myosin. Now there are some regulatory proteins which regulate the process of contraction. These include tropomyosin and troponin, which has three subunits, that is T, C and I. In addition, in order to provide structural stability to the muscle, there are anchoring proteins. Some examples are titin, desmin, dystrophin and actinin. Let us now study the different components of the sarcomere. Now let's first look at the contractile protein actin. So actin is in the form of thin filaments which are anchored to the Z line of the sarcomere. There are two types of actin, that is G-actin or globular actin and F-actin or filamentous actin. Most importantly, there is only one binding site present on actin, and this is to bind myosin. On the other hand, if you look at the contractile protein myosin, it is the thick filament which is anchored to the M line in the center of the sarcomere. Now, the shape of myosin is very characteristic. It is filamentous in nature, and it has a body, a tail, and a head which projects sideways. So the projecting head is referred to as the cross bridge. Now, this cross bridge has two binding sites. One is for actin, another one is for ATP, which provides the energy for muscle contraction. So this is how the myosin molecule looks, quite like a cobra with a projecting head. So this is the tail portion, there is a small body, this is the neck portion, and this is the head. So this entire thing, the head portion, is actually projecting sideways, so we call this as the cross bridge. You can see that it has two binding sites, one for actin and one for ATP. Let's now look at the regulatory proteins involved in muscle contraction. Now broadly, there are two important regulatory proteins. One is a long filamentous protein called tropomyosin. So you can see this green molecule, that is tropomyosin. So the role of tropomyosin is to cover the myosin binding site of actin. And this is why at rest, actin cannot bind to myosin. The other regulatory protein is troponin, which is made up of three subunits: troponin T, troponin C, and troponin I. Now, troponin T, as you can see, is bound to tropomyosin. So the function of troponin T is to ensure that the tropomyosin is covering the binding site of actin. When there is muscle contraction, the same troponin T becomes activated and removes the tropomyosin from the actin molecule. Troponin C binds to calcium during muscle contraction. So when a muscle has to contract, calcium is released and that binds to troponin C. Troponin I is an extra precaution. So in case the myosin binding site of actin is exposed for any reason at rest, the troponin I binds to this site and inhibits the actin. So these are the two important regulatory proteins. Now let's come back to the sarcomere, which is the functional unit of the skeletal muscle. So under the light microscope, if you view the skeletal muscle fibers, you'll see that the myofibrils are made up of repeating units called sarcomeres. And these sarcomeres give the appearance of alternate dark and light bands, and that is why the muscle fibers look striated under the microscope. Now you can see the sarcomere here. The boundaries of the sarcomere are formed by the Z lines. Now to these Z lines, it is the actin filaments which are anchored, and these are relatively thin. So we also call them as thin filaments. So this region appears light under the microscope, and it's called the light band or the isotropic band, represented by I. So the I band is made up of thin filaments. Now you can see that in the center of the sarcomere is a line called the M line, to which the thick filaments or myosin is anchored. So this region appears dark under the microscope, and it is called anisotropic band or A band. So this is made up of the myosin filaments or the thick filaments. Now within this A band, you see that there is a relatively lighter zone, and that is because in this region you do not have any actin overlapping the myosin. It is only myosin. So this region appears little lighter than the region in the neighborhood. So this is called the H band. So we already saw that the boundaries of the sarcomere are formed by the Z lines, and in the center of the sarcomere is the M line. And we have already defined the sarcomere as the region between two Z lines, and it is the basic functional unit of the skeletal muscle.